Hello, everyone. My name is Mike Connolly. I'm a fifth-year Ph.D. candidate in Dr. Nikki Traylor-Null's lab at the University of Miami Rosenstiel School of Marine and Atmospheric Science. My Ph.D. research is focused on understanding interactions between Pocillopora corals, innate immune systems, and their associated bacteria communities. And today I'll be talking about some results from experiments that exposed Pocillopora corals to immune stimulation with bacterial lipopolysaccharide, as well as treatments with broad-spectrum antibiotics intended to disrupt the normal bacteria community. So corals are important because they build the coral reefs that support immense biodiversity and invaluable ecosystem services throughout the tropics. However, corals are also popular models for the study of host microbe interactions. And this is because in addition to the well-studied endosymbiosis between corals and dinoflagellate algae, the family Symbiotinaceae, corals also live in persistent association with a diversity of bacteria that are now being understood to benefit the health of the coral holobiont through various mechanisms, including carbon fixation, nitrogen cycling and amino acid synthesis, sulfur cycling, and antioxidant functions, as well as B vitamin provisioning and the production of protective antimicrobial compounds. So my research is focused on the widespread and resilient genus Pocillopora, which also possesses a complex and diversified innate immune system that is involved in mediating these host microbe interactions and possesses several classes of pattern recognition receptors, signaling pathways, and effector mechanisms. And my research involves the use of immune stimulants and broad-spectrum antibiotic treatments as experimental tools to activate coral immune responses and disrupt coral immune microbiome interactions, and then measure the resulting effects on coral gene expression and bacteria community shifts. So for my experiment, I traveled to southern Taiwan and collected Pocillopora corals from two sites, Hoan and Wanlitung. I collected 2 Pocillopora colonies at each site, and after sequencing, these would turn out to be 4 genotypes from 3 different cryptic species. Hoan 1 was Pocillopora demicornis type 8, Hoan 2 was P. demicornis type 4A or type alpha, and at Wanlitung, both genotypes were Pocillopora acuta. And this distinction is important for how we interpret the bacteria community and gene expression data. So I subjected replicate fragments of these corals to my experimental treatments, including the controlled treatment and ambient temperature seawater. I then exposed fragments to bacterial LPS at 10 micrograms per milliliter for 4 hours. And I also exposed fragments to the broad-spectrum antibiotics ampicillin and streptomycin for 72 hours. I completed this design using 3 fragments of each genotype in each treatment for a total of 36 fragments, and then preserved samples at the end for bacteria community and RNA-seq analyses. So I found that LPS exposure activated Pocillopora immunity, but that overall transcriptome patterns were genotype specific. There were 395 differentially expressed genes, and upregulated genes included toll-like receptors, immune-related transcription factors, and pore-forming toxins such as perforin 2. Downregulated genes included ammonium transporters, antioxidant enzymes, and a few neuropeptide receptors. Similarly, Pocillopora transcriptome responses to antibiotics treatments were also genotype specific, and there were 730 differentially expressed genes. I've added treatment centroids to this plot to help visualize the treatment effect. Interestingly, when I then aligned the non-coral reefs to the Cladocopium genome, I found that the algal symbiont response to antibiotics treatments was greater than the host response, with 1,614 differentially expressed genes. The processes involved in the antibiotics response in the host included upregulation of intracellular trafficking secretion and vesicular transport genes and post-translational modification, protein turnover, and chaperones, with a downregulation of genes involved in inorganic ion transport and metabolism. In the symbiont response, downregulated functional classes of genes included energy production and conversion genes and inorganic ion transport and metabolism genes, suggesting that interfering with the coral-associated bacteria community negatively impacts the metabolic exchanges occurring within holobiont. My 16S sequencing results show that antibiotics treatment has a clear effect on the coral bacteria community. Furthermore, the dominant bacteria taxa that are driving these clustering patterns belong to the families Rhodobacteriaceae, Ultramonidaceae, and Endozoicomonadaceae. And these results altogether show that we can use antibiotics treatments as an experimental tool to cause shifts in coral bacteria communities and then assess the resulting effects on coral and symbiont health. So you'll see here that the Rhodobacteriaceae are nearly completely eliminated by antibiotics treatment, and bacteria in the family Ultramondidaceae increase in relative abundance to occupy that vacant niche. So with that, I want to thank my advisor, Dr. Nikki Traylor-Knowles, and my academic committee at Miami of Dr. Mike Schmally, Margie Alexiak, Andrew Baker, and Alex Wilson. I'd also like to thank Andrew Mayfield and the folks at the National Museum of Marine Biology and Aquarium in Taiwan for help completing this experiment, as well as my co-authors P.J. Liu, Crystal McRae, and Ray Tan. Lastly, I want to acknowledge my funding sources, the University of Miami Graduate Student Fellowship and the NSF East Asian Pacific Summer Institute Fellowship that sent me to Taiwan, and the Rasmus David Rowland Fellowship that funded the sequencing experiments. Thank you, and I'll be happy to discuss any questions with you in the Q&A.